In OncoLand, we can search for multiple genes at the same time, which allows additional comparisons between these genes. In this example, we will search for KRAS, TP53, and CDKN2A. When multiple genes are searched at the same time, additional views become available. The default view shows the frequency of mutations of either KRAS, TP53, or CDKN2A, but we can see the individual mutation frequencies by alteration distribution by gene view. We can see that KRAS is almost always mutated in PAAD tumor samples, but CDKN2A and also TP53 are frequently mutated. We can find the co-mutation frequencies under DNA-seq somatic co-mutation frequencies. Filter under tumor type for PAAD. Note that these genes are frequently co-mutated. Another convenient way to view co-mutation frequencies is with the alteration omic print. Each block is an individual sample, and the color indicates mutation status of that sample for each gene. "55 samples have mutations for KRAS and TP53 and either a mutation or deletion of CDKN2A. Expression of each gene can be displayed in a single chart with multi-gene variable view. Correlation of expression between genes can be shown with the multi-gene correlation view. Fusions between a pair of genes can also be analyzed. In this example, we will search for TMPRSS2 and ERG. In Select View, choose RNA-seq fusion, fusion gene pair frequency. In PRAD tumors, fusions between these genes commonly occur. Select the fusion browser to directly observe fusion locations. A split window is shown, with the genomic location of TMPRSS2 in one window and ERG in the other. In tumor samples with the gene fusion, several ERG exons have much lower exon coverage. Below, you can see each unique fusion event identified as a circle. Clicking on a circle in one pane will highlight the corresponding circle in the other pane, indicating the fusion location. Additional information will also be displayed about the fusion.